Hello, everyone. I'm Johanna. I've been working at the Sports Center for Computational Neuroscience as a postdoc for a couple of years, and now I'm a senior researcher and team leader at Thunder Labs, which is a German neurotech startup. And I'm gonna talk about importing, cleaning, and pre-processing data today. So why do we need to pre-process our data? So when you measure EEG data and it comes out of the recording device, it's like measuring a difference of potential on an oscilloscope. So to make sure that we can use the data and we can make sense of the data, we need to extract meaningful measures from it, for example, brain oscillations and brain source activations, compare brain data in different conditions, and assess reliable changes through stimuli, for example, ERPs. But before we can do all that, we usually apply a series of transformations to the data. So first, we need to import the data to the software that we are using to analyze it. And so how to do that, it always depends on the toolbox and the data format. But it's kind of a problem that there are so many different data formats and also ways to annotate the events, for example, record information on the data sets. So there have been efforts to establish a new standard, how to store your data, which is the brain imaging data structure. Bits. So it's not a data format, but it's a way to organize your data to make sure that the documentation is consistent and complete. So basically, it's like a file structure where you save your data sets, and there are files with descriptions on the data, like the channel locations, for example, or which reference you use. Where the data was recorded and things like that, and event table, for example. And this is important to facilitate the reuse by others and also yourself. So I think that everyone knows the situation, for example, when in your lab you have some data that was recorded a couple of years ago, and maybe you want to reanalyze it, but it's really hard to find information, and then it takes a long time to get all the information together, or it's even impossible, and then you have to kind of throw the data away. And also currently there are there's a lot of effort to publish data online, to make it freely available. And for that, it's also really important to have a consistent and complete documentation of what was happening in your experiment and the information related to the data. BITS is not a new file format or a search engine or a data sharing tool. And there have been, since a couple of years, I think the MEG paper was from 2017. So first BITS was for fMRI data and MRI data, but MEG and EEG have been included. Almost all the open source tools have tools for bits. So to import bits and import data in bit format or export data in bits format. EEG Lab and I think EEG Lab and Brainstorm, they have dedicated tools for processing group level data. So basically you can, if you have like a data set with 20 subjects or something, in bits format, you can directly import the whole study in EEG Lab or in Brainstorm and do group analysis on that data. Then the next step would be then, once you have imported the data, you need to make sense, you need to import the event markers. And that's another problem because often event markers are not properly documented. So sometimes they're just even numbers. So yeah, and maybe it's like a one word or something that you know that is a button was pressed or something like that. So there have been developments using hierarchical event descriptors, HEAD tags, that have also been developed at the Sport Center, and they're also included in bits, so you can use it in those files that you store with bits. And it's basically a way to annotate your data or the events. So for example, once you already recorded the data in EGLab, there is a tool for creating head tags where you can associate, for example, some terms, different keywords with your stimuli, just to make it more clear if someone ever wants to analyze it, reanalyze it, or also to make it machine actionable. So if you want to use automated pre-processing pipelines and AI, for example, it's basically impossible if you don't have those annotations. And it's also trying to create like a universal language of the events. So for example, you have like one universal word for, I don't know, sensory stimulus or something like that. So you don't need to hard code it in your code once you analyze the data, but you can basically, you know, write a code for analyzing like a bunch of different data sets. So yeah, in practice, then you could, for example, take from an online database, like here in NEMAR, it's a curated database for EG data that is connected to high performance computing cluster at the University of California. And basically you could then here have those different experiments with a bunch of different subjects, and then you can select the head events, and then you can use automated pipelines in those toolboxes to do your analysis, and this also makes, for example, meta-analysis over several studies possible. Yeah, so once you import the data, you also need to import channel locations and labels. So sometimes maybe you don't have any locations, and then you have to use standard locations, which is suboptimal. But scanning electrode positions will become standard. So in the past, it was kind of more difficult because it was expensive to buy those devices to measure the electrode positions. But now you can basically do it on a smartphone or a tablet, and you basically just scan the head of the person. Currently, it's like that. For example, in EGLab, you still, once you have the image, someone still needs to go over the image to click on each electrode to record the location. But currently, EGLab and field trip teams plan to work together to automatically align scans with templates, and then the manual electrode labeling process is speeded up. Okay, so once we have imported the data and the event markers and channel locations, we are usually doing a couple of transformations on the data. And these pre-processing pipelines are usually, yeah, they're either people develop their own pipeline out of experience or through recommendations from others, or yeah, some recommendations, for example, from your own lab or from other people. But it's good, I mean, it's good to have some kind of objective measures as well, like what kind of pre-processing parameters you want to use. So Arno Delorme, this is a recent paper where he basically tested different pre-processing parameters and also different pipelines in all the open source toolboxes that are presented in the workshop. And his outcome measure, so he tested it on 3 different data sets. One was, this one is that where people were looking at animal targets and non-animal distractors. Then the phase data set that we're going to use, the Wagner and Hansen data set, we're going to use in the workshop where they were looking at familiar and scrambled phases, and then just a regular oddball task. And basically the outcome value, so the was the number of significant channels in that time area. So it was around like between 300 and 500 milliseconds after the stimulus. And yeah, so I'm gonna go through the different pre-processing steps and then show some outcomes of this analysis that Arno did. So let's go to first to re-referencing and downsampling. So when you reference your data, I mean, there are many different options. You can use earlobes, the nose, mastoids, for example, average reference. But yeah, so we should remember that the location of the reference matters. So if you, for example, have your recording electrode here and the reference here, then the potential that you record will be negative, and if it's the other way around, it will be positive, because you are measuring the difference between those two electrodes, basically. Yeah, it also changes the shape, the polarity, and also the amplitude of your ERP. So this is a study where they compared different ERPs for the presentation of phases, so different references. The green one is the nose reference, the red one is left mastoids, and the blue one is average reference. So basically, I mean, you can see that here. So when the reference is at the nose, then of course the signal that you record at the nose is 0. But you have like a positive potential for the left mastoid. But then in the parietal electrodes, for example, the ERP looks similar for the average reference and the nose, but completely different for the left mastoid, for example. If you want to use source localization, usually it's best to, you should always use the average reference. This paper here, for example, they also tested different preprocessing parameters, and they also checked average reference and mastoids for the error-related negativity and the flanker task. And they basically showed that here, you can see in this gray area, that the average reference is the amplitude of the ERP, and for the average reference is a little bit smaller than for the mastoids. So like, because negative would be larger here. But then when you look at the standard deviation unit, so the between trial standard deviation for the ERN, and then you can see that the average reference actually reduces a lot the inter-trial standard deviation, which means that kind of the quality of the data is better because you don't have outliers. It makes the data more uniform. Yeah, then let's look at Arno's analysis. So he compared a lot of different references, and this is just for 2 of the data sets. So this is the oddball data set and the phase data set. Basically, like the nose is kind of worse for the oddball data set here, which is probably also sometimes because the nose is inducing a lot of noise. But the other references pretty much all do similar, and they are compared to using no reference. We don't know why using no reference here is superior to using a reference. So basically, if you use a reference, it increases the number of significant channels. Yeah, but we can look at the, at the comparison, and yeah, average reference usually does pretty good. Then you might want to downsample your data. So why would you want to downsample your data? Because maybe you want to reduce time and the computational cost. Because if you have data sampled at 1,000 hertz, for example, and you want to run ICA, it takes much longer, of course, than if you have data sampled at 200 hertz. And most MEEG processes are below 100 hertz. So maybe there's no need to keep a sampling rate at 500 hertz. But of course, you always need to be aware of the Nyquist frequency. So if you want to measure or look at processes that are at like 150 hertz or 200 hertz, you need to have at least double the sampling rate. And reasons for not lowering the sampling rate are that even with MEEG amplifier noise, for example, information above 250 hertz might be useful for some algorithm, also for detecting muscle activity or something like that. And also behavioral responses are measured on the order of milliseconds, so maybe you want to keep your data at the same time scale. Usually, if you record your data at 500 hertz, it's enough for most EEG and MEG analysis. The next step, you need to filter the data. So one important step is to high-pass filter the data because there might be slow drifts. So for example, if you look at the raw data, here there are slow drifts in the data, and then once you apply a high-pass filter, this is 1 hertz, then the data looks much nicer. But in this comparison on the ERN amplitude and the standard deviation, the high-pass filter has little influence on the ERN amplitude and the inter-trial standard deviation, but a baseline is used here. So the baseline might also bring the epochs to the same level, basically. Yeah, Arno also did some analysis on the high-pass filter, and it actually shows that, so you should use at least the high-pass filter at 0.5 hertz, so this is doing best. If you have only one at 0.0 hertz, 0.01 hertz, then the number of significant channels is much lower. And then he also tested different filters in the different toolboxes. So we have the EEG Lab FIR filter, Brainstorm FIR filter, and MME FIR filter. And they all do pretty much the same compared to the ERP Lab Butterworth filter, which is an IR filter. The only filter that does much worse is the one in field trip on epochs. So you should really filter your raw data and not the epoch data. Then let's look a second at the baseline. So once you already did your pre-processing and you extracted epochs and you are subtracting a baseline from the data. So this comparison basically showed that long baselines, or the baseline doesn't really have any influence on the amplitude of the ERN, but it reduces the trial standard deviation if you use a long baseline. So long baselines are usually better. And that's also what Arno found in his data. So, so if you use a long baseline, it actually gives you more significant channels. Basically in all the data sets, but there is no significant difference to having no baseline at all, which is probably because he high-pass filter the data. So if you have a good high-pass filter, you don't really need to use a baseline for epoching. Then there was another paper where they were looking at minimal high-pass filter with a short baseline. So they used 0.01 hertz and a baseline of 100 milliseconds. And then Arno compared those values to just using a high-pass filter at 0.5 hertz and not using a baseline. And he actually found that using the high-pass filter without baseline is much better than using this minimal high-pass and the short baseline. So yeah, so this minimal high-pass filter and the short baseline is reducing the number of significant channels a lot. So this would always be kind of like the gray line that it was compared to. So after filtering the data, we want to examine the raw data just to look at the data, how it looks, because it might be that we just recorded, noise. I don't know, maybe the reference was not properly attached or something like that. So we want to look at the raw data, and we also want to look at artifacts. So we have different kinds of artifacts. So for example, low frequency, eye movements, linear trends. We have high frequency events, which is like here, for example, the muscle activity. And we have here short discontinuities in the data. Then we want to identify and reject bad channels. So you can either look at the raw data for bad channels and try to identify it yourself, but it's also, I think when you start working with EEG, that's maybe like a good strategy, because just to get used to how the data looks like. But if you have, let's say, 40 subjects, it's really cumbersome to go to 40 subjects to look for the bad channels, and then it also might be kind of subjective to choose, which channel to reject. So it's good to have some objective measures, which are the same over subjects and maybe also over studies, to reject bad channels. So one way of finding bad channels is, for example, to use pairwise correlations. So usually in good data, channels that are close to each other on the scalp, they should be highly correlated with each other, and in bad data, that's not the case. Yeah, then Arno, he also kind of tested what interpolating bad channels does to the number of significant channels in the data. And he found that interpolating bad channels does not increase significance for reasonably clean data. So we also have to say that those data sets that he used, they're very clean, so there's not that much noise happening. The only data set where it basically improved the results was for the oddball data set, but the other ones, there's basically no difference to no channel correction. And what about line noise? So if you remove and interpolate, channels that have a lot of line noise, and here for at least two of the data sets, removing the noisy channels that have line noise, that improve the number of significant channels. But if you just notch filter the data or use line noise removal, it doesn't improve it. I'm not sure why it's like that, but I think that sometimes it's also when you have a lot of line noise in one channel, is sometimes because the channel is not properly, has not proper connection with the scalp. So it's picking up a lot of noise. So removing those kind of channels might really, improve your analysis. And then we want to reject large artifact time points. So you can look, just at the raw data and maybe reject parts of the data. Or you can use an automatic method to be a bit more objective. For example, there is artifact subspace reconstruction in EEG lab. That is basically computing PCA on windows of data and comparing it to some reference clean data that is selected during the cleaning process. Yeah, and then you can it's basically subtracting that activity from the EEG, and then you can, you can use it either to remove those birds, or you can also reject those, like the portions of the data. Yeah, Arno, again, he compared it for the different data sets, and he found no gain using ASR, but he used only the option to reject the portions of the data. And yeah, the data is very clean again. But it also seems that the removal of bad trials doesn't compensate for the loss of statistical power, because then you have a lower trial count. So you have less trials. You have less trials to get a significant result, basically. And then he also checked in Brainstorm, MNE, and Field Trip, and other automated rejection methods in EEG don't show any gain either. Yeah, and then we can also, for example, run ICA and reject components. So yeah, you can have artifact rejection methods where you reject portions of the data, so you can completely get rid of the part of the data that has the artifact, or you can correct the artifact where you just remove the artifactual activity, but you keep basically the portion of the EEG without the artifact. And one way of doing that is with independent component analysis, and this is a systematic review that looked at which artifact rejection correction methods people are using, and the most used method is ICA, which is here 34%. And then 20% are using hybrid methods, so ICA was something else. So yeah, yeah, just short on ICA. I'm sure that everyone already heard about it. I mean, the underlying idea is that the EEG is basically a sum of currents that are recorded at the scalp electrodes. And ICA is a linear data decomposition method that can decompose those linearly mixed signals and recover the true source signals, and through that also separate artifact sources from brain sources, basically. So what's important here is also the quality of the decomposition. So if you have a very bad ICA decomposition, then there are probably a lot of still artifact and brain signals mixed at the scalp, so you cannot really, get rid of the artifacts by rejecting components, because you will have both brain and artifact sources mixed. A good ICA decomposition usually has components where the scalp maps look dipolar, like here. And when you have something like this, which looks more like, I don't know, like a psychedelic plot, then yeah, it's probably not good. Yeah, so the idea is that you have your EEG data. So you have these are the channels, and you can see that there is, for example, eye blinks, which are in almost all the channels. present in almost all the channels, even in the central ones. And once you run ICA, you can separate all those different sources. So basically, you have the EUG, 2 different EUG components, and then you have, for example, EMG here, and ECG, which looks very nicely separated here. And then you have also the brain components there. The problem is usually once you run ICA, how do you select the brain components? And that's also kind of like a really subjective. So there are efforts not to automate this process. So you have an automated method how to select brain and artifact components. And there is, in EEG-Lab, there is a new method, IC label, which classifies components into brain components, muscle components, eye components, heart components, I think, and then there are others, and channel noise. And it's really quite reliable, so it's working really well. And they also have a website, so for collecting labels, but also it's like a tutorial for practicing labeling of components. So if you want to, improve your skills to label brain or artifact components, you can go to this website here, and you can try it out and then see if you're correct or not in your labeling, component labeling. So there are several automated ICA classification methods. Most of them just basically classify some kind of artifact. So some they only classify EOG components, so to detect EOG components. Some only detect muscle or heart. I think IC label is the only one that can classify brain components, and also like the other artifact components. And you can also try all those methods, so they're all implemented in EEG-Lab, so they're all EEG-Lab plugins. But for example, the other toolboxes like Fieldtrip, MNE, and Brainstorm, they can apply ICA, but they don't have any automated classification methods currently implemented. If you use MEG, you cannot use those methods because they are trained on EEG data. So if you want to have an IC label for MEG, you could start a project to retrain IC label for MEG data, which I think would be a good thing. So let's look at those comparisons again. How ICA improves ERN amplitude. So it doesn't improve the amplitude of ERN, so it doesn't increase the amplitude. But it reduces a bit the trial standard deviation. So it means that the quality of the data is better. So it's kind of removing, extreme values. And Arno also tested this, so he looked at the significance of the channels again, and he found that, for 2 of the data sets, it doesn't change anything, so it's not really, helping you to get more significant channels, except for this data set where they were looking at the animal images. But we need to also think that ICA may remove artifacts which contribute to differences between conditions. So maybe the number of significant channels is not always the best metric to use, or, we should also kind of look at different metrics, how it's changing the data sets. So it's not ideal for assessing the performance. So should I even use ICA to remove artifacts then? To make sure that the condition differences you are you found are based on brain activity, not artifacts. For example, eye movements often contaminate ERPs, and yes, in some experiments, people might always blink, for example, when they see an error. So it might really change the ERP that you're looking at. And then it's also removing channel artifacts in sculpt topography, so it's good for visualization. And it also may improve sequence source localization. Yeah, so here, for example, if you see the sculpt maps of the raw data and then the high-pass filter data, yeah, it still has some channel artifacts, and the data then cleaned with ICA and IC label. Yeah, the sculpt maps look really nice. But we also need to think that this data is very clean, so people were just sitting in front of the computer. The experimenter was really, you know, careful that people are maybe not moving their head or not clenching their teeth or anything. But what happens if you go to more realistic scenarios, for example, in mobile brain body imaging, I mean, you need, you have much more artifacts. And this is an example, for example, where they were measuring visual evoked potentials during walking and running. And here you can see really a big difference, you know, before and after cleaning with ICA. So before, these are the sculpt maps, and the red lines are basically the ones before cleaning with ICA. So in the background, you can see that it's really variable and the activity is really big. And the gray lines are the ones after cleaning with ICA. And yeah, you can see that here it really makes a big difference because the signal can be completely buried in noise. So, I mean, the benefit of using ICA also always depends, you know, on your data and the application that you have. And for example, here, we recorded people walking in a gait robot, and we run ICA, and then you can see, for example, like if you just keep brain components or brain plus physiological noise or just the raw data, and these are basically the spectra at the different channels. And you can see that, I mean, the signal can be completely buried in noise, because if you don't remove any components, you will have like a spectrum that is flat like this. Yeah, and this was published 2012 in Neuroimage. I just forgot to put the reference here. So yeah, Arno, he also compared all the different pre-processing pipelines in the different toolboxes. So he compared EEG Lab, MNE, Brainstorm, and Field Trip. Basically, the outcome was that compared to high-pass filter at 0.5 Hertz, there is not much improvement of any pipeline, although the EEG pipeline is the only one significantly superior to 0.5 Hertz high-pass filter for all three data sets. Yeah, the assumption is that EEG Lab has a minor advantage due to line noise, contaminated channel rejection, and ICA automated rejection. You can see here in the top panel, these are basically the number of rejected trials. So yeah, if you have more rejected trials, it of course decreases, you know, your statistical power, and then you might have less probability to get significant results. All those automated pre-processing pipelines are available on GitHub. So you can test your data with the different pipelines and look, you know, what are the differences. And you can comment and help improve these pipelines for each software. And there's also the EEG Lab YouTube channel, where you can watch videos on those, how to apply those pipelines, and they will be online soon. So yeah, and the toolbox maintainers have been given the opportunity to comment on the videos. Yeah, and the videos will be released in January, so you can look at them, you know, and try it out yourself. And that's it. Thank you.
Thank you so much, Johanna, for this very complete and interesting talk. We have time for one or two questions if you have any. Otherwise, we're kind of running behind schedule and we will move to the tutorials. But anyone wants to ask a question right now? Yes, there is one here.
Hello, thanks for the talk. Just a short technical question. When you do a high pass with a low frequency, you usually have a pretty long filter. But then if you have pretty strong artifacts that happen quickly, applying this filter would actually propagate this artifact further. So my question is whether there's some consideration for this and how one can handle it. Like, I mean, if you have a channel that looks more or less like that in the raw data, and if you filter it, this like spike, it will propagate for the entire filter duration that may be of like 1 second or even 2 seconds, depending how low this frequency is.
But I think that's more for low pass filter, right? I think the high pass filter is not propagating the artifact, should not propagate the artifact.
If it's long, it still should, no?
Yeah, and it's usually, I think if you use FIR filter, it's also usually not that, but I think that usually the high pass filter shouldn't propagate the artifact. It's usually, I think, the low pass filter that's kind of like smoothing it over time. But you could also, like, if you see that you have like a very bad channel, for example, that is really out of range, you could completely remove it. And I mean, the filter is only propagating the artifacts for that specific channel. So if you would remove it anyway, then you will not have any problem at the end.
Another last question out there. Okay, thanks.
Thank you. The measure you took to assess the efficiency of the different methods is the amplitude of an ERP component. So I assume it's because the ERA is difficult to get. Has this same type of thing been done for more time frequency analysis? So is there any resources? Is it the same, the effects of the different pipelines and analysis? Or is there any specific resources for that?
So I think for time frequency analysis, it's not honestly like the filter, for example, is not that crucial because you will anyway look at the data in those just in those specific frequencies. But actually, I don't know if there are papers that are looking at that. Yeah. Okay, thank you.
Okay, I think we have to move on. Thank you again, Johanna.